There are many couples in the world that want to start families. However, sometimes genetic disorders can get in the way of that. Consider Sarah. Sarah knows that Huntington's disorder runs in her family, and she may carry the gene mutation that puts her at risk of developing the disorder when she's older. Huntington's is a very severe disease that usually leads to the patient's death when they're older. Sarah, knowing that she could pass this disease on to her child, could take the risk and conceive a child normally. But Sarah wants to know what her other options are. So she goes and meets with a genetic counselor. The counselor informs Sarah of a process called Preimplantation Genetic Screening, or PGS. PGS is a method that works in conjunction with in vitro fertilization. The embryos are genetically screened for the disease before uterine implantation. That way, healthy embryos can be specifically selected for. PGS can be used for diseases that are caused by mutations in single genes or for major chromosomal disorders. In Sarah's case, the embryos would be screened for the repeat expansion mutation that causes Huntington's disorder, and only an embryo that contains the healthy Huntington's gene would be implanted.
For more information on the procedure, Sarah goes to meet with a doctor. She explains the process of PGS. First, the ovaries will be hyperstimulated with hormones so they produce more eggs than normal. These eggs, or oocytes, will be removed from the ovaries and then in vitro fertilization will occur. Oocytes that were successfully fertilized will be grown in the lab for 3 to 5 days. At this time, a few cells will be microsurgically removed from each embryo, and the embryos will be frozen while genetic testing takes place on the cells. One of the methods used to analyze the DNA is PCR, or polymerase chain reaction. The first step is denaturing the double-stranded DNA. Then, primers complementary to the Huntington's gene are used to bind to each strand. These primers allow a polymerase to attach to the DNA and add complementary nucleotides to each strand, creating a double-stranded DNA fragment from each template. This process is repeated until the DNA fragments reach a high enough concentration. Then, the DNA fragments can be analyzed using gel electrophoresis, which separates DNA based on size and charge. Each amplified segment of DNA is inserted into the well of an agarose gel. Then, an electric field is applied to the gel and the negatively charged DNA migrates towards the positive end. The mutated Huntington's gene has extra repeats of a particular nucleotide sequence, leading affected segments to be slightly heavier and therefore migrate down the gel slower. Once embryos without the mutated Huntington's gene are identified, 1 to 2 will be taken out of the freezer and implanted in the uterus. Sarah can then choose to save the rest of the frozen embryos for a future pregnancy.
Now that she's learned about PGS, Sarah has to decide if this is the option she wants to choose. There are several drawbacks she has to consider. First, despite the higher rate of success of PGS, there is still a chance that the mutation could go overlooked. Thus, prenatal and newborn screening will still be necessary to look for diseases that the fetus could be carrying. Also, PGS is a rather invasive procedure on both the mother and the embryo. There is always the chance that other issues could arise and the PGS would not be successful. However, despite the complications, PGS can significantly lower the risk of passing on Huntington's disorder to her child, which makes it a worthwhile choice for Sarah. She and her partner decide to go through with Preimplantation Genetic Screening.
Although an embryo with a healthy Huntington's gene is implanted, Sarah still undergoes regular prenatal screening. Prenatal screening tests are generally non-invasive and analyze either the mother's blood or an ultrasound for genetic abnormalities. Prenatal screenings can only tell you the risk your child has of having certain conditions. If one of the screenings shows a high risk for certain abnormalities, prenatal testing can be done to make a more certain diagnosis. Luckily for Sarah, the prenatal screening results look favorable and she has a healthy pregnancy. Once her child is born, doctors conduct a standard newborn screening to test for a wide array of disorders. Her son tests negative for all of them. Sarah's son grows up healthy and is not a carrier of Huntington's disease. Sarah's son will not develop Huntington's disease, and he won't have to worry about passing it on to his children either, all thanks to Preimplantation Genetic Screening.